Annual plant propagation is different enough from the perennial plant propagation, so it's warranted that we spend some time talking about those differences and some of the nuances related to it. The main difference between perennial and annual propagation is that you use seeds for annuals for the most part. Of course, you can use seeds for propagating perennial plants too. Cuttings and other things that we've discussed so far are faster often, and they're successful too, to use cuttings and divisions and layering and grafting and things. But so you can use seeds for that, but when you're dealing with annuals, almost all of them are done with seeds. And so knowing how to do seed germination and about the seed anatomy and things like that are helpful for seed germination of annuals. I know that there are some exceptions to that. People talk about being able to have tomatoes grow perennially, especially in the Southern California area, and that you can actually take cuttings of some annual plants and do rooting of the stem cuttings of those. But something that an annual plant is a plant that lives its entire life within 1 year. So it seed sprouts, it matures, it flowers, it gets pollinated, and it sets seeds all within 1 year. So taking cuttings and rooting those, it's okay, but you're spending a couple of months just getting those stem cuttings off an annual. Imagine a tomato plant, taking a stem cutting off a tomato plant and spending a couple of months getting it to root and then planting in the ground. It isn't really buying you a lot of, there's not really a lot of benefit from that compared to just growing it from seed. So most annuals are done. They're propagated by just collecting and germinating new seeds of those same plants. Here are just some basics on the seed structure of plants. You've got the seed coat, also known as the testa. Cotyledon are the embryonic leaves, so those will become the first two things that are somewhat leaf-like that pop out when that seed germinates, emerges from the soil. Think of a peanut. Those two halves of the peanut are the two cotyledons of a peanut. Have you ever noticed on a peanut there's a little nub kind of at the bottom in the middle? That's the little embryo that would become the new plant that has the new leaves, the real leaves. They're embryonic at that stage, and called the plumule, and a part called the hypocotyl, which is right underneath those embryonic leaves, which will become the new stem, and the radicle, which is the embryonic root. So you've got these two big kind of cotyledons, like on a peanut or a kidney bean. They have these two halves. Those are the part of the seed that gets pushed up above ground, and they produce, they provide the nutrition for the growing seedling before that seed has produced real leaves. Once it produces true leaves that can photosynthesize, then it can make its own food, but the cotyledons are the nutritional source that comes in the seed itself to get it to the stage when it can produce new leaves, true leaves. Almost all seeds have some type of dormancy. So this is another one of those things that you need to learn about if you're using seeds for propagation. In dormancy, the metabolism of that tissue, of the seed itself, falls. It goes down, and the number of organelles, so mitochondria, chloroplasts, vacuoles, things like that, all the things that are normally in a cell that keep it vital and functioning and metabolizing, those decrease. So the cell becomes very simplified and slows down, and the water content in that cell decreases quite a bit. It becomes dehydrated. The vacuoles, which are gas bubbles, somewhat like a bubble inside the cells, which have functions. It's not just a bubble. They deflate, and the food reserves become more dense. They crystallize inside the seeds. So it just slows down and simplifies and shrinks. And the reason we want to talk about and know about dormancy in seeds is because when we plant seeds in propagating annuals, we want to be aware of their dormancy and aware of how to break that dormancy. to germinate the seeds. There's physical barriers that keep the seed dormant, like the seed coat. It keeps oxygen and water from getting into the seed. And you already know some of this, like when you plant a seed, then you start watering it, and or if you drop some seeds into water, they will, if the seed is able to, the seed will take in water and expand. So that seed coat of a seed that you did that to, like if you dropped a kidney bean in some water, it was able to expand because the seed coat was thin enough that it was permeable to water. So if a seed is in dormancy, that means that somehow it's got some layer that's keeping water from getting in and oxygen also from getting in to the seed. So there's a physical barriers. The seed itself is dehydrated, so adding water is, and getting the water being able to get into the seed is going to break the dormancy. And there's also chemical inhibitors that are present sometimes inside seeds that keep that seed from germinating. There are things called growth promoters that are around inside the plant when the plant is growing and germinating, and the dormancy, when the seed is in dormancy, those growth promoter molecules are absent. Those are all the things inside the seed that maintain the dormancy. Seed viability is another thing to know about. A seed is viable when it will germinate if the environmental conditions are met. So if it has enough water and oxygen, sometimes they need light or warmth. Those conditions are met and it germinates. When those conditions are given to that seed, then it was a viable seed. In general, seeds last for 10 to 15 years. Actually, some of them last a lot less than that. Like short-lived willow seeds that last only about a week, less than a week, and some can last for over a century or more, like mimosa seeds. There were some seeds found in Egyptian tombs that were over 2,000 years old, some rice grains, and those germinated, so that's a real extreme example. But the conditions are important for how long it will last, how long it will stay dormant. Very dry, cool, dark conditions will keep most seeds viable for, it'll maximize their viability. Now, genetically, they can only stay viable for so long. And if you want to, ensure that it stays viable for the longest that seed type can, cold, dry, anaerobic conditions are what will maximize that. So let me walk you through the steps of dormancy breaking. So to get a seed to germinate, these are the steps you need to know about that goes on with a seed. So you've got a seed, let's say it's in the soil. First, the barriers, the physical barriers have to be broken down. The seed coat has to become permeable to oxygen and water. So that seed could be rolling around in the soil for a while, gophers shove it around, roots push it, microbes decompose a little bit microscopically, weaken the seed coat. There's abrasion, fire could come by and also weaken the seed coat, and that's the barrier that's broken down. And then water gets absorbed, given there becomes some kind of moisture in the soil that seed is in, it starts to rehydrate, absorbing water. And then there's inhibitors inside, there's growth inhibitors in the seed. Those start to be destroyed because of the water that's been imbibed, and light, warmth, and water also further break down those inhibitors, and then you get production of the growth promoters that get that seed to start growing into a seedling. Some more on the germination steps. So you've got pre-germination, rehydration, and water being imbibed or taken in by that seed. A little more detail on the steps. RNA and protein synthesis is then stimulated, so it's starting to have cell division occur. And the metabolism increases. Remember, it was highly decreased in the dormant stage. And food reserves are starting to be broken apart and releasing their energy. And cells start to change in actually their structure. They start to expand, and the ultrastructure, meaning kind of like the the infrastructure within a cell that keeps its shape and function and allows it to metabolize and be a fully functioning cell. You get more organelles and things like that starting to fill the cell up again. And then it starts that little seed starts to actually germinate. The seed coat is ruptured by the growing embryo. The emergence of the seedling, usually the radicle, the thing that becomes a root, goes down first. And then you see often the little neck of the hypocotyl pushes up and pushes the cotyledons into the air. And then you get more root growth, and the hypocotyl expands to be the new shoot or stem. And you start to get true leaves that are formed, and then you start getting photosynthesis, and the food storage tissues like the cotyledons start to senesce or wither away. So at this point you might be thinking, oh my, we've been talking about some pretty straightforward techniques and kinds of methods and data as far as plant propagation goes with the asexual means, and now we're getting all nitty-gritty in the biochemistry of the cells and tissues of seeds. That's kind of the nature of propagation of plants by seeds, is that you have to know about the dormancy and know about these biochemical changes that are occurring in seeds. You don't have to know all the details here, but these are really some of the basics, just understanding dormancy and then understanding how to break it and what's going on in the cell after dormancy is broken. I used to work for the Forest Service, and I did a lot of native plant propagation, and every single species had its own method of being propagated. And we did almost all of our propagation was by seed. We would go into the forest, which was actually high desert, gather seeds from all these different native plants, and then grow them in the greenhouses. And we needed to know a lot because every seed would be treated in different ways. Some would need cold treatment, some would need hot treatment, some seeds would need fire treatment, some would need smoke treatment, and other chemical treatments to break their dormancy. So that's why we're going into all the details. So the food reserves are broken down through the process of hydrolysis, and growth hormones start to be produced inside the plant as it's growing. There's one called gibberellin, and its counterpart is a growth inhibitor, which is abscisic acid. And those are two hormones that control growth and inhibit growth. So there's differences in how the plant is being developed. Those different hormones and inhibitors directly affect the genes for enzyme synthesis and the activity of enzymes themselves. And the growth substances are affected by environmental factors like light, temperature, and humidity. So all of those things relate to these food reserves. A little more on seed dormancy. The dormancy is really important for seeds, as I mentioned before. It allows the seed to germinate when conditions are favorable. So if you think about it, if you're a tree and you produce your seeds, most trees are starting to grow in the spring, a shrub too, or any kind of plant. And they start to produce flowers and they produce seeds. You're getting into late summer, fall, and then the seeds are mature in the fall. If all those seeds get dispersed or fall to the ground, and then those seeds have no method of having dormancy, then they'll sprout right then, right before it gets really cold in a lot of parts of the world and the United States, and have freezing temperatures in the winter. So all these seeds that just sprouted, then you get a frost or a freeze or a snow, all those seedlings would die. So seeds go dormant, so they go dormant until the conditions are favorable for them to grow. Not just conditions favorable for them to germinate, but to germinate and then grow into a plant that will survive more than a year or two. So hopefully that makes sense to you. We're talking a lot about seed germination, but again, you wouldn't want your seeds, if you were a tree, the seeds that you're producing, let's say acorns. You don't want those acorns to fall and germinate right at the time when they're going to have to experience a cold snap, and that will kill the new little oak seedling. So dormancy is important. It creates viable seeds they're still viable seeds if they're dormant, they just don't germinate, and they're waiting basically for the conditions to be right. If it's too cold, the conditions aren't right. Dormant seeds are regulated by environmental factors, so factors in the environment, like moisture. A dormant seed needs sufficient moisture and the right temperature and adequate soil oxygen levels and light. Not all dormant seeds need light, but some of them do. And when the seed is exposed to sufficient moisture and warmth and oxygen, then it will germinate and hopefully grow into a full-fledged plant. Germination, so how does the seed know when it's moist and warm and high oxygen levels? It has ways to know that, and those conditions themselves will break the dormancy. The moisture and the temperature affect those seeds biochemically to make that seed able to germinate. There are two main ways to get the seed to break the dormancy on the seed, and that is to soften or weaken the seed coat, and that's done through what's called scarification, or break the internal, sometimes called endogenous, so internal dormancy, is to stratify, or using the process of stratification. So again, germination is inhibited by the seed itself, that's the dormancy, and it can be broken by breaking the external dormancy, that's the outer, like physical seed coat, keeping it from getting water and oxygen inside itself. You can weaken that seed coat through scarification or scarifying. It's kind of like wearing it down, abrading it, cutting that seed coat, making it permeable, or breaking the internal dormancy through a stratification, which is treatment with hot or cold temperatures. Let's first talk about seed scarification. So that's when you're weakening the outer seed coat. That's when water and media, so if you took some seeds, let me back up, took some seeds and put them in some good media and added warmth, and the seeds still won't germinate, it may be that it needs extra scarification. You need to weaken the seed coat even more. A lot of like the wildflowers you buy in little packets at the store, or like poppies, or those kind of fun little seeds for your garden, or vegetable seeds, they do have dormancy in them, but the dormancy is broken immediately upon exposure to moisture and a little bit of warmth. So let's say you're planting some basil and some tomatoes. You get some of those seeds at the garden store. They're dormant. They're not sprouting inside those packets in the store. They have dormancy. But as soon as you plant them in the soil and they're exposed to moisture and warmth, they sprout. So their dormancy is easily broken by giving them those environmental conditions. But some seeds, you plant them, and you give them the warmth, and you give them the moisture, and they still won't germinate. So scarification is then necessary, like actually active additional scarification, and that's making the seed coat permeable to water and gases. It's a natural thing that happens from freezing temperatures or microbial activities in the soil where the seed is, that crack and fracture and weaken the seed coat, but it takes time and kind of exposure to those conditions. It's not just going to happen by planting them. They have to, sometimes the seeds have to pass through a digestive tract of an animal that eats the seed or the fruit that was surrounding the seed, and excretes it, and then the digestive enzymes and acids that were in that animal's gut have weakened the seed coat. So when they defecate that seed out, then now it's ready to germinate if it's given moisture and soil and some heat. So us humans, if we want to scarify seeds to increase germination, we can mechanically weaken that seed coat. We can file a nick in really thick, hard seed coats, like a lotus seed, something like that. You can take a file and cut a nick through the seed coat just until you hit the internal part of the seed, the cotyledons or endosperm. And then you can plant it, and that allows water to get in and gases, which allows the whole process of pre-germination and germination to unfold. You can sandpaper seeds on the outside, that weakens the seed coat, cut them a little bit with a knife, crack them gently with a hammer. Hot water treatment can scarify them. I've taken seeds and put sand and seeds into a rock tumbler and tumbled them for like a week, and that abrades the seed enough so it is able to take in water and oxygen. There's also chemical weakening of seed coat. You can soak your seeds in a diluted sulfuric acid. Sometimes you can also do vinegar or even lemon juice, and that can be enough to weaken the seed coat chemically. Of course, be very careful if you're using sulfuric acid, and wash your seeds after you've soaked them in those acidic substances, whichever one you choose. Completely wash them afterwards so that acid isn't sitting on their seed coat any longer. Again, scarification is making the seed coat permeable to water and gases. The seeds, after you've treated them, should look duller in appearance. That means that they're more pockmarked. Microscopically, there's holes and fissures, and the water and oxygen can get in. Let's next talk about seed stratification, breaking internal dormancy that are regulated by the inner seed tissues. So again, the seeds that have internal dormancy are meant to be that way to prevent the seed from germinating when the environmental conditions are not good, where the seedlings they might germinate, but they wouldn't survive. So two types of internal dormancy. There's shallow, which mostly disappears with dry storage, like a lot of vegetables and flower seeds. Like I said, you get in little packets. You don't need any special treatment to overcome that kind of dormancy because, again, just planting them breaks that. There's also a deeper type of dormancy where the seeds won't germinate until subjected to a particular duration of moist, cold or moist and warm temperatures. So think of it this way: you're putting your seeds into wet, like in maybe a mixture of wet media or sand, and putting them in the refrigerator for a couple of months, or putting them in a warm, some kind of type of warm chamber for a couple of months. So in most of our temperate zone or temperate climate plants where we have cool or cold seasons, the seeds of those plants need to go through a cold stratification, where they are, if we're trying to get those seeds to germinate when we want them to, then you need to give them a cold treatment by, usually the refrigerator would be a fine temperature. Refrigerators are about 36 to 42 degrees, and that's good for mimicking a winter, so environmental conditions of a winter, which those seeds internally know they need to go through before they germinate. But we can hasten that process by subjecting them to not as long as they would need, like a full winter, but usually like 30 to 90 days of cold stratification will break that internal dormancy. Here's just a few details on how you would do that. There's also warm stratification, seeds that need warm temperatures and a treatment just like the cold stratification, but using putting them in the conditions where it's 68 to 86 degrees Fahrenheit. There's also double dormancy. The seed coat, the external dormancy and internal dormancy is involved for that seed. And some species have that double dormancy. And first you would scarify it, weaken the seed coat, and second you would stratify it, give it some kind of cold or warm treatment of that seed, and then it is able to germinate. Some examples of seed dormancy. Oak acorns do not go dormant, if you didn't know that. When planted in the fall, the roots will emerge. During the winter, they start to grow right away, and the shoots will emerge in the spring. So if you want to keep acorns longer than normal, cold stratify them just to keep them from germinating through the late fall and winter. They won't germinate if you put them in some wet sand in a Ziploc in the fridge, and then when it comes time in the spring to plant them, you can take them out of that. Usually they start to sprout just a little bit inside your fridge in that bag, because they don't go dormant, but they slows their metabolism down. So if you want to wait to plant them until it's a good good springtime warm conditions, then it's better to do that. But otherwise, they can sprout in the right in the fall. So, and sometimes that's good for seeds, and sometimes it's not. So if you're going to be propagating plants, let's say you're propagating some oaks for somebody else, and they want like 300 oaks for, who knows, their nursery or a property, you would want to do this cold stratification to hold them over until the spring, so you've got all of them to germinate and survive. The redbud, western redbud, Cercis occidentalis, germination is inhibited by an impermeable seed coat and embryo dormancy. So one recommended treatment is soaking for 30 minutes in concentrated sulfuric acid or vinegar, then followed by 3 months of cold treatment or cold stratification. And you can also use mechanical scarification too instead of the vinegar or acid treatment. And again, whenever you're going to do some type of dormancy breaking on a seed or propagation of an annual or propagation of a perennial, but from seeds, just look up as much as you can about the techniques that are most successful for that species. And you'll see some of the similarities, and you can form your own methods that you try. And talk to people if they've propagated it from seed before. They're going to be the best source of information on the actual details of the methods that work the best. Maple trees vary in their requirements for seed treatment to break dormancy. Some maples require some type of treatment and some don't. There's a lot of maples, like the red and silver maple, which are pretty common. When I lived in Ohio, they were all over the place as street trees, and some wild too. But the seeds would come down in the late summer and fall. They're little whirly-gig like helicopter seeds floating down everywhere all over the roof, and then they'd land and accumulate in our house's gutters and germinate. So I'd have to clean out the gutters in the late fall because they'd be full of all these little baby red maple and silver maple seedlings. So they didn't have any dormancy really, or it broke just when they got in some kind of moist situation. Some of our maples, like the big leaf maple that lives in higher elevations in the coastal ranges here in California, they do require stratification for a couple of months using fresh seeds, so they have more dormancy than the ones out in the Midwest, and they do need some kind of treatment. So it just depends. Again, you'd have to find out with each type of seed you're going to be germinating, find out the dormancy breaking requirements of that particular species. Some pines require treatment, like southern pines and loblolly pine have almost none, have no treatment that's needed, no dormancy. They can just take them out of the cones and plant them. If you grew up around dormancy breaking requirements for pine seeds also vary by species. Some things like southern pines or loblolly pine, which are really common in the southeast, but also some of those are planted as landscape trees in the southwest here. They don't have any kind of dormancy requirements. I often remember seeing seeds falling out of the cones in our trees in the Bay Area, and then popping up in our lawns later to get mown down when we mowed the lawn, but no dormancy. But some other types require 30 to 60 days of cold stratification to germinate well. So some of them will germinate without that stratification, but if you want all of them or a really high percentage of them to germinate, then cold stratify them. And there are some pines like the Tory pine, which does really well when it's fire stratified, so it's adapted to open its cones during high intensity heat. And then the seeds themselves having to go through some of that fire, and the seed coat is weakened during the fire and the smoke and the heat, and then they're able to germinate. So again, varies by species. And now let's get into talking about the seed sowing. So instead of the technical things about trying to break dormancy, a lot of the seeds are going to have a similar type of treatment after their dormancy is broken or during that time. You want to put them in shallow trays or flats. They don't need a lot of soil at first to germinate. Their roots are going to be small at first, so you can use minimal amounts of media to sow them into. Plugs, these fabricated types of soils that are made of peat or rock wool, they can be used also to put seeds in. In general, the rule of thumb is about 2 times the diameter of the seed is the depth that it should be planted. Cover it with a little bit of media. Maintain moisture and humidity near the soils. Put it in a warm place. Partial shade is best, but doesn't always required. You've planted seeds outside that germinated and grew okay, but for a lot of these perennials and some annuals, not your normal garden vegetables, but other types might need a little bit more protection from bright light when they first germinate. There are lots of different methods for sowing seeds into the ground. You want your soil to be soft and friable, means lots of small particles, loose, and free of big chunks and debris. Weeds, no weeds. Broadcast seeding can be done or put into rows or along borders. Rake the seeds in, protect them from birds, rodents, and cats. You don't want seeds to be eaten up right away, especially from birds or rodents. When you plant them, you want them to be, sometimes you can soak your seeds overnight in water or put them into moist soil where they're protected, maybe inside, so they take in water before they're actually planted outside in the ground where when they're dry is when rodents and birds tend to want to eat seeds. The animals and chickens and birds and things tend to be adapted to find and eat dry seeds. That's their food source. But when those seeds have absorbed water, their smell tends to go away. Rodents and birds aren't able to find them as well, and the seeds have a higher chance of germinating. Of course, keep the seeds moist and warm after you planted them, and when they start to germinate, if there's too many and they're too crowded, you can pull them apart or thin them and plant them into the permanent spot that you want them to grow into maturity. If you're collecting seeds from a plant in the wild or in your garden, make sure that you remove everything off of the seed before you store them. You don't want any kind of tissue or chaff, something that's kind of the outer husk of the seed. You don't want that staying on there while it's in storage because it could have moisture or some kind of pathogen on it, which could later damage or create disease in the seed itself. So you want to clean them as much as possible. Chaff is all the organic material that's not the seed that surrounds the seed, like the ovary wall or the seed capsule, and it can contain, like I said, fungus, bacteria, insect larvae, which can degrade and kill the seed later. Think of like a tomato. If you want to save tomato seeds, you can take out the pulp that was inside a tomato that you really liked from your vine, and you can take those seeds, but there's all that soft, squishy pulp all over the seeds, so you can't just put that in a Ziploc and store it. It would rot. You usually take that pulp and you spread it out on some kind of paper towel or paper plate, and then let it dry completely, and then you pick off the seeds once they're dried and all of that pulp has been consumed by bacteria. So make sure they're clean. You don't want any of that kind of sugary or nutritive chaff or tissue remaining on the seed when you store it. And overall, baby plants, seedlings, need 6 important things to help them germinate and mature into a nice small plant. They need moisture through your irrigation system. They need warmth, some kind of heating source like a heating pad or a actual heater inside your greenhouse or home. Sterile media, shade or partial light provided by a shade cloth or a greenhouse structure. Protection from what? From pests, from the elements, etc. And ventilation, fans that keep the humid air moving. Humid air that's not moving is really good for bacteria and fungal infections, but moving humid air that's moving pretty good by fans is going to be good for the plants and not encourage too many fungal or bacterial outbreaks. So there you have it.